Welcome back on Botany for You online platform. I, Dr. DIPA CHAUHAN, is going to discuss phytochrome interacting factors. In nature, there are several environmental factors like soil, water, light, pH, temperature, humidity, moisture. All these environmental factors play some role in the life of living organisms. Light is such an important environmental factor. We know that light is essential for the process of photosynthesis as plants trap solar energy and convert it into chemical form of the energy. Light also plays important role in the life cycle of all living organisms, especially in plants. Light controls growth and development. The light results in physiological and development responses in the life cycle of plants. For example, germination of seed, flowering, the initiation of flowering, the number of flowers, the number of flower primordia, growth of plants towards light are some of the developmental physiological responses of plants towards light. Light induced development are termed as photomorphogenesis, where plant growth patterns respond to the light spectrum. There are two types of responses of plants towards light signals. One is phytochrome-mediated responses and another is blue light responses or cryptochrome-mediated responses. Responses of plants towards light are mediated by phytochrome, and phytochrome acts as a photoreceptor. It absorbs red and far red light region. It also absorbs blue light region of the spectrum. Phytochrome exists in two photochemically interconvertible form. One is the PR form and another is PFR form. PR form absorbs red light and PFR forms absorb far red light region, and both are interconvertible. PFR form is physiologically active form. The current slide shows absorption spectra of PR and PFR form of phytochrome from etiolated Avena seedlings. The PR shows absorption spectra 660 nanometer and PFR shows absorption spectra 730 nanometer. PR absorbs red light and is converted to PFR, and PFR is converted to PR by absorption of far red light. The table shown on this slide shows morphogenic responses of the plants towards red light. For example, germination of seed initiates in lettuce, detiolation takes place in oat, flower growth is inhibited in xanthium, and there are so many responses enlisted in this table. For example, gymnosperms. Gymnosperms too show responses towards the light. For example, uh, exposure to red light stimulates chlorophyll accumulation in pinus. Pterophytes do show stimulation of growth. That is, in young gametophyte on exposure to the red light, the initiation of growth take place. Bryophytes too show replication of plastids. Stimulation of replication of plastids take place, and rotation of chloroplast is a response seen in green algae. Phytochrome activation leads to photoresponses, and it is visually observed in the form of some morphological or developmental changes. It has some time, lag time, to observe those responses, which ranges from few minutes as well as to few weeks. Effects induced by red light are irreversible by far red light only for a limited period of time. Now, certain amount of light is required to induce responses, and the amount of light which is required to induce responses is termed as fluence. So fluence is defined as the number of photons per unit surface area. And the lag time as well as escape time differs in diverse responses. There are three types of responses, which is based on their sensitivities to fluence. The three types are very low fluence responses, low fluence responses, and high irradiance responses. Very low fluence responses are non-photoconvertible and they are initiated by very low fluences. Example of such very low fluence responses is stimulation of growth of coleoptile and inhibition of growth of mesocotyle in oat seedlings. Low fluence responses needs minimal fluence and they are photoreversible. For example, luteus seed germination, which belongs to this category. High irradiance responses require long exposure to light of relatively high irradiance. The examples are synthesis of anthocyanin, production of ethylene in sorghum, flowering induction in hyoscyamus, enlargement of cotyledons in mustards, and these are non-photoreversible responses. Another type of responses is cryptochrome-mediated responses. Photoresponses in plants are controlled by blue light also, and they are believed to be mediated through cryptochrome. Cryptochrome also acts as photoreceptor. Phototropism, stomatal opening, inhibition of hypocotyle elongation, phototaxis, chloroplast movements within cells, and synthesis of carotenoids, chlorophylls are example of such cryptochrome-mediated responses. The graph over here shows absorption spectrum for blue light in Avena coleoptile, which leads to stimulation of phototropism. Three peaks are indicating the action spectrum of blue light. Phytochrome. As we know, by this time, phytochrome acts as photoreceptor. It detects lights in plants, bacteria, and fungi. It was discovered by Borthwick and Hendrick for the first time in 59. Phytochrome is a chromoprotein and it occurs as a dimer. It is made up of two subunits and each subunit is of 125 kilodalton. The subunit consists of two components. One is the light absorbing pigment called as chromophore, which is having a linear tetrapyrrole. Another is the apoprotein. The light-absorbing pigment molecule chromophore is attached to the apoprotein. The attachment is through thioether linkage. So we have the structure of phytochrome over here. We have two subunits, the light-absorbing pigment called as chromophore and the apoprotein. The phytochrome has two terminals, C-terminal and N-terminal. The protein part comprises two identical chains, A and B, and each chain has a pass domain, GAF domain, and Phi domain. This pigment can exist in two forms, one that absorbs red light and one that absorbs far red light. Phytochrome also act as temperature sensors as warmer temperature enhances their deactivation. And we know that phytochrome FR is the active form and it leads to the responses within the plants. The phytochrome FR leads to germination, de-etiolation, inhibition of stem and petiole elongation, chloroplast development, leaf expansion, branching, flowering, senescence. These are some of the responses enlisted due to the activity of phytochrome FR. Phytochromes controls several aspects of plant development, which has been listed over here. Phytochrome FR is physiologically active form of phytochrome. Conversion of PR to a PFR by light produces particular responses, and the responses depends on the localization of phytochrome and state of differentiation of the responding cells. Phytochrome, which act as phytoreceptor, and the first step is the absorption of light by the pigment. Then the absorbed light alters the molecular properties of phytochrome, which induces a sequence of cellular events, ultimately leading to a growth in ultimately leading to change in growth, development, or position of an organ. The effect of phytochrome is concerned with the PFR effect on changes in the properties of cellular membranes. Photostationary state ratio, that is PFR ratio to phytochrome total, acts as a signal pursued by the plant under certain conditions. For example, 3% PFR level is necessary to induce HIR response. We know that light regulates growth and development in the plant, and it is visualized in the form of responses. The visualization of responses takes place at two stages in the plant's life cycle, stage of seed germination and seedling development, and at the time of flowering. The photomorphogenetic responses are classified into two. One, the type 1, that is fast responses, which include those processes in which light energy absorbed by the plant is transduced to another form of energy. Example is movement of life in mimosa. Ion fluxes, chloroplast movement. Type 2 responses are relatively slow and it occurs on a time scale of hours and days. For example, stem elongation, germination of seed, opening of the hook, expansion of leaves, flower initiation, and biosynthesis of pigments. In higher plants, 75% of the total amount of phytochrome is localized in protoplast. 25% is equally divided between vascular and epidermal tissues. PR form is distributed diffusely and PFR form is present in a discrete localized pattern. Phytochrome PR is stable protein, whereas PFR can bind and fuse to membrane, thus modulating the activity of membrane. In contrast to higher plants, type 1 responses in simple organisms are mediated by membrane-bound phytochrome. For example, movement of chloroplast in Mogacia is membrane-bound phytochrome response. Phytochrome regulates plant gene expression by absorbing red and far red light. A basic helix loop helix transcription factors called phytochrome interacting factors interact physically with the red and far red light phytochromes. Phytochrome interacting factors are transcription factors that act as repressors of photomorphogenesis. Their inhibition by PHIs leads to substantial changes in gene expression. PIFs act as key regulator of the transition from scotomorphogenesis, that is, growth in dark, to photomorphogenesis. PIF function as negative regulators of light responses by repressing photomorphogenesis and maintaining the scotomorphogenic state of the etiolated seedlings in darkness. Upon exposure to light, phytochromes promote the turnover of PIFs through rapid phosphorylation, ubiquitination, proteasome mediated degradation, which inhibits PIF function to induce transcriptional reprogramming, resulting in photomorphogenic development. In Arabidopsis, we have 8 phytochrome interacting factors. This module is the phytochrome PIF signaling module. It illustrates the phytochrome and PIF interactions. The connecting light with various colors illustrates the interaction of each phytochrome with different PIFs. For example, phytochrome A interacts with phytochrome F1, phytochrome F3, whereas phytochrome B interacts with phytochrome F1, F2, F4, F5, F6, F7, and F8. Same way, we have rice. In case of rice, phytochrome A interacts with PIL, phytochrome C interacts with PIL, and phytochrome B interacts with PIL. In case of non-vascular plants, for example, moss, liverworts, we have various phytochrome interacting factors. For example, in case of liverwort, we have one phytochrome interacting factor which interacts with MP5. Phytochrome interacting factors have been discovered in variety of plant lineages. For example, Arabidopsis has got 8 phytochrome interacting factors. Phytochrome interacting factor 1, phytochrome interacting factor 3 to 8, and phytochrome interacting factor 3 like 1, PIL1, which is renamed as PIF2. PIF2 have an APB motif. In rice, there are 6 phytochrome interacting factor like proteins with the conserved APB motif. In case of Arabidopsis PIFs, the overexpression of pills also promotes hypocotyl and internode elongation. In case of maize, 7 phytochrome interacting factors are there. Phytochrome interacting factors function primarily as negative regulators of photomorphogenesis. In addition to their prominent role in regulating photomorphogenesis, PIFs have shown to regulate many other pathways, which include metabolic signals to the circadian clock, thermomorphogenesis, hormone signaling, and biotic-abiotic responses. Regulation of photomorphogenesis reflects the shared or overlapping functions of PIFs. PIF also function distinctly to modulate certain physiological responses, which are controlled by a single PIF. PIF1 plays a major role in inhibiting light-dependent seed germination. It also regulates chlorophyll biosynthesis, development of plastids by modulating the expression of number of genes is done by PIF1. PIF2, which is also known as PIL1, regulates seedling de-etiolation, and this is the positive regulation in response to blue, red, and far red light. PIF2 has got all the amino acids which are essential in the basic domain and is predicted to be a DNA binding HLH protein. PIF3 represses chlorophyll biosynthesis and photosynthesis in etiolated seedlings. It also interacts with TOC1, and this interaction optimizes the temporal regulation of diurnal growth of hypocotyl elongation. PIF4 regulates hypocotyl elongation in response to light, shade, temperature, and diurnal conditions. Besides elongation of hypocotyl, PIF4 also controls several developmental and physiological aspects, for example, stomatal development in response to the quality of light, circadian rhythms, chlorophyll degradation, senescence in darkness, tolerance towards freezing situations, anthocyanin, biosynthesis under red light conditions. All these things are regulated by PIF4. PIF5 functions as a positive regulator of leaf senescence through chlorophyll degradation in darkness. It is a negative regulator of red light induced anthocyanin biosynthesis. PIF5 also functions at the interface between red light signaling and the circadian clock through its direct interaction with TOC1. PIF6 produces two splice variants, alpha and beta, of which beta form regulates seed dormancy. Contrary to the general role of PIFs as negative regulators of photomorphogenesis, PIF6 function as a positive regulator and it inhibits hypocotyl elongation under continuous red light. PIF7 acts as negative regulator of seedling de-etiolation under red light. PIF7 along with PIF3 and PIF4 functions additively to promote elongation of hypocotyl under continuous red light by suppressing phytochrome B levels. It also regulates shade avoidance responses by directly controlling auxin biosynthetic genes under shade conditions. Functional importance of FI8 is largely unknown. Phytochrome interacting factors are known to be degraded under light, but PIFs are also actively degraded in darkness. Thus, a homeostasis of phytochrome interacting factor level is necessary for the proper transition from scotomorphogenesis to photomorphogenesis. Now, to adapt to the surrounding environments, plants constantly monitor and respond to changes in the red and far red regions of the light spectrum through the phytochrome family of photoreceptors. Extensive efforts using genetic, molecular, and photobiological techniques have led to the identification of a group of basic helix loop helix transcription factors called the phytochrome interacting factors, which directly bind to the photoactivated phytochromes. Members of phytochrome interacting factor family have been shown to control light-regulated gene expression directly and indirectly. Phytochrome interacting factor 1, 3, 4, 5 are degraded in response to light signals and physical interaction of phytochrome interacting factor 3, with phytochromes is necessary for the light-induced phosphorylation and degradation of PIF3. Phytochrome interacting factors constitute an excellent model for the investigation of the biochemical mechanism of signal transfer from photoactivated phytochromes and the light regulation of gene expression that controls photomorphogenesis in plants. Thank you. These are the references.